Hi, folks. How you doing today? Okay, welcome back, everyone. I hope you're doing well. And, we're approaching that weekend time, so that's nice to get a little bit of a break. Okay, so we have a busy day today between lecture and lab. It's all going to be about the cell cycle, mitosis and meiosis today. Okay, so lots to do. But before I get started, do you have any questions you want to ask at this time? Okay, all right, not hearing anything. Let me just ask in the chat window. Okay, are you alive out there? All right, okay, good. All right, nice to see some responses there. Okay, all right, so folks, let's get started. And, feel free to ask questions along the way. So last time we were together, we got a decent start into talking about the cell cycle. We were talking about the somatic cell cycle, so the version for reproducing body cells. And I brought in this little diagram, which is similar to things you're seeing in your textbook. We talked a little bit about cell cycle arrest, which is G0, and explained that there's some legitimate reasons why a cell might enter a non-proliferative state. And then we proceeded to talk about the various stages of the cell cycle, G1, S, G2, and we also outlined and commented on some cell cycle checkpoints. Okay, so those are things that we talked about from last time. So I'm going to get set up to continue this conversation, moving into the specific nuclear events of mitosis. Okay, so okay, let's get this rolling here. And what we have here is cell cycle continued, chapter 2. Okay, so let me ask just a couple questions in the chat window to see if, how you're doing with your personal understanding. Okay, so let me ask this in the chat window. How many chromosomes would be present in a human body cell during G1? So gap phase 1, how many chromosomes are going to be present? What do you think, folks? Okay, thanks, Maddie, for the participation. I deeply appreciate that. Anybody else? Okay, all right. So so the consensus for the answers in the chat window seem to be 23, and that is incorrect. Okay, there are going to be 46, right? There are going to be 46 chromosomes present in a G1 cell. I'm talking about cells of your body that are part of your body cells. They're not reproductive cells. They're, the cells of your skin, cells of your heart, cells of your brain, cells of your liver. You're going to have 46 chromosomes present in these cells. Here's another question for you. How many chromatids are present in a human G1 cell? How many chromatids are present in a human G1 cell? Okay, thank you, Jade. Anybody else? Okay, all right. So we might maybe have some disagreement here. Okay, and this is the reason I'm asking these things, because we need to hash these things out. And today in lab, we're going to do some things to try to clarify what's going on here. But the answer to the question is 46 chromatids. Okay, so there would be 46 chromatids in a human G1 cell. Okay. And the reason for that is because each individual chromosome is made of one chromatid. Okay, so 46 chromatids in a human G1 cell. Each of the 46 chromosomes is made of one chromatid. Let me ask another question. How many chromatids will be present in a human G2 cell? How many chromatids will be present in a human G2 cell? Okay, thanks, Jelena. How about anybody else? Okay, all right. So, we have a little bit of disagreement here, but in fact, the answer would be 92. And that's well, so there's 92 chromatids present in a G2 cell. That's the correct answer. Okay, 92 chromatids present in a G2 cell. Here's my question for you. How many chromosomes are present in a human G2 cell? So the last question was how many chromatids are present in a human G2 cell. Now I'm asking you how many chromosomes are present in a human G2 cell. Thank you, Anthony. Anybody else? Okay, is everybody confident that Anthony's correct, or you're just not sure? All right, well, to move things along, Anthony is correct. There are 46 chromosomes in a human G2 cell. Okay. And the reason it has 46 chromosomes but 92 chromatids is because each of the 46 chromosomes is made of 2 chromatids. Each of the 46 chromosomes in a G2 cell is made of 2 chromatids. Okay, human G2 has 46 chromosomes. Each chromosome in a G2 cell is made of 2 chromatids, making 92 chromatids. Okay, so here's my last question, then we're just going to lecture for a little bit. Why does the G2 cell have 92 chromatids? Why? Okay, so Jade's responded. How about the rest of you? Okay, so Jade is correct. Thank you, Jade. She's absolutely right. The G2 cell is going to have 2 chromatids per chromosome because during the synthesis phase, the S phase of the cell cycle, that period between G1 and G2, the DNA was copied and hopefully copied correctly, okay? All right, so start thinking about these things, and I'm going to start pushing these issues more deeply in lab this afternoon. But okay, so let's talk about, so we're talking about mitosis at this point. Okay, we're going to talk about mitosis at this point. All right. Okay, so you know that from our conversation last time, and forgive me popping back and forth, but we've covered G1S and G2. So now I got to talk to you about mitosis, okay, the events of mitosis, which are also shown to you in a pictorial version in figure 2.7 of your textbook, figure 2.7. Okay, so mitosis refers to the events that happen inside the nucleus of a cell that wants to copy itself, inside the nucleus of a non-sex cell that wants to copy itself. Okay, so before I go any further, here's an important idea. Okay, question. What is the purpose of mitosis? Well, I'll answer that, right? The purpose of mitosis is to create genetically identical cells from a precursor cell, okay? To create genetically identical cells. And so the purpose of creating genetically identical cells would be to allow your body to heal itself if you've been damaged. So if you fell down outside and scraped your knee on the macadam, right, that's going to smart, that's going to hurt. But give it a couple of days, you'll form a scab and it will heal over, right? So the cell cycle interphase plus mitosis allows the skin on your knee to heal. At the same time, mitosis allows you to make more cells so that you can physically get larger, so that when you were 3 years old, you were 2.5 feet tall. But when you're 18 years old, you're 6 feet tall, right? So it allows you to grow up. So since we're talking about this, maybe we should ask, what is the purpose of meiosis? Just so we have that comparison point and contrasting point. So the purpose of meiosis, as you probably know, is to intentionally create genetic variation in cells so that the next generation of human beings that are produced from you are not genetic clones, okay? So the purpose of meiosis is, I would say, twofold. One, to intentionally reduce the cell chromosome number by half. And secondly, to intentionally introduce genetic variation in those chromosomes. Okay, so doing these two things that we've documented in your notes allows your body to produce sex cells, which allows us as a species to produce children, and it ensures that our children, while they might have similar features, characteristics, and traits to us, are not clones of us, right? So you're not having an exact replica of yourself walking around that's 2.5 feet tall. That would be kind of odd. That would be kind of weird. Okay. So if that wasn't in your mind, hopefully it is now in your mind. So when we talk about these processes, you have that in the background. Purpose of mitosis, create a genetically identical cell. Purpose of meiosis, create a genetically distinct cell that allows for the species to reproduce and create children. Okay. All right. So before I start describing mitosis, I think this would be a good time to show a video on this because, you know, probably a 4-minute cartoon is worth like 1,000 of my words very easy because it's just nicer to see this. So what I'm going to do is show you a number of things today. And the first is this video on cell cycle and mitosis. I'm going to put the video in your Mathematica file so that you can review it again during a private study time. And I'll also remind you that in Mastering Genetics, in the study area, there's a section called BioFlix. BioFlix also has a video for mitosis and a separate one for meiosis. You might want to check those out. So just remember, Mastering Genetics, study area, BioFlix for mitosis and meiosis videos. Okay. All right. So let's watch this first video on cell cycle and mitosis. Okay. So just give me a second here. Okay, so I'm going to get optimized here so that the sound comes through to you properly. Okay, and here we will go. Please enjoy, and you can watch this again during a private study time.
Adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S, or synthesis phase, the DNA replicates. Finally, during G2, or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Okay, folks. So, I apologize. It seems like the resolution on that video was not as good as I would have liked it to have been. But if you do use the BioFlix video in Mastering Genetics, hopefully you get a much cleaner image. So let's go through the stages of mitosis and document some of the major features of this process. You might want to have figure 2.7 up in your textbook at this time. Can be a useful visual guide as we go through this. Okay, so let's continue here. Okay, so the stages of mitosis. All right, so first is going to be prophase. Okay, so you can see visibly what prophase kind of looks like in figure 2.7. This is one area of your textbook where it really does have some nice images for you to look at. So let's talk about this a little bit. So prophase highlights. Okay, so several things happen during prophase. As the cell moves into prophase, the disorganized and fairly more dispersed chromatin material is actually going to take on a discrete shape. It's going to take the noodle of spaghetti or pasta that we talked about last time, and it's going to intentionally wrap around proteins into what's called a nucleosome. And we'll talk about nucleosomes in another chapter, but suffice it to say, the chromatin condenses into the chromosome. Okay, and we know, because of things that I tickled you with in the chat window, that these chromosomes are made of dyads, meaning that each chromosome has two sister chromatids, and the sister chromatids should be genetically identical because the purpose of mitosis is to create genetically identical cells. So if DNA replication during the synthesis stage has been faithful in what it is trying to achieve, we should have two molecules of DNA, two chromatids called a dyad making up each chromosome, and they should be genetically identical. Okay, so let me ask a question here, and I'll put this question into your notes as a way of stimulating your thinking. So as I ask the question in your notes, if you could type in an answer into the chat window, I appreciate that if you think you know the answer. Okay, so here comes the question. How many nucleotide pairs of DNA will be in a human non-sex cell during prophase? How many nucleotide pairs of DNA will be in a human non-sex cell during prophase? Okay, so remember the size of the human genome, which is a statement about the haploid content of a typical cell, would be 3.1 billion nucleotide pairs of DNA. So how many nucleotide pairs of DNA will be in a human non-sex cell during prophase? Thank you, Megan. Okay, thanks, Jade. How about the rest of you? Okay, so, okay, thanks, Juanita. All right, so to keep things rolling, we'll just keep going here. So drum roll, the correct answer is 12.4 billion. So why is that the case? Let's think about that, okay? Remember, body cells, and I say body cells, I'm specifically referring to cells of your body whose job it is not to make sperm or egg. So non-sex cells. Remember, body cells start out as diploid. They're already diploid. Why? Because that body cell ultimately came from 23 chromosomes from your mom and 23 chromosomes from your dad, right? A long time ago, before you were ever born. So remember, body cells start out as diploid, meaning they have 46 total chromosomes. Okay, so that represents the 2n equals 46 condition. 2 copies of every chromosome. Chromosome number 5 from mom, chromosome number 5 from dad, chromosome number 17 from mom, chromosome 17 from dad. You have 2 copies. So we have 2n equals 46. So those 2n equal 46 copies in a G1 cell, you would have 6.2 billion nucleotide pairs of DNA. But because the G1 cell had to go through the S phase of the cell cycle and copy its DNA before it could get to G2 and now prophase of mitosis, means that the DNA has actually doubled. So we've gone from the diploid state of 6.2 billion nucleotide cells to a diploid state where I still have basically quadruple the content of a haploid cell. Okay. So remember, the body cell starts out as a diploid, 2n equals 46 chromosomes, containing 6.2 billion nucleotide pairs of DNA. A prophase cell, like I'm asking you about in this question, will have copied its DNA during the S phase of the cell cycle. So its DNA content will be 12.4 billion nucleotide pairs of DNA. Okay. All right. So it's a little bit weird to think of it this way, isn't it? So think of it. A sperm cell, an unfertilized egg cell, they are haploid. N equals 23 chromosomes. Among those 23 chromosomes is 3.1 billion nucleotide pairs of DNA. In a fertilized egg cell, which goes on to produce your stem cells, which then differentiate into every other cell of your body, they're going to be diploid. So they're normally going to have 2n equals 46 chromosomes. So they're going to have 3.1 billion times 2 nucleotide pairs of DNA, or 3.1 times 2 is 6.2 billion nucleotide pairs of DNA. So your diploid cells of your body have 6.2 nucleotide pairs worth of DNA, billion nucleotide pairs worth. All that DNA is copied during the S phase of the cell cycle. So you're going to go from 6.2 to 12.4 billion nucleotide pairs of DNA for a cell that is in the prophase stage of mitosis. Okay. All right. So I know this can be confusing, which is why I'm asking the questions. So let me ask you this. Are you okay right now? Would you like to ask any questions about this? Is it making sense, folks? Please give me feedback, because I can't see you. I don't know whether you understand or whether you don't understand. Okay. All right. Okay, thanks for the feedback. I appreciate that, because I hate not being able to see you. I mean, I just have no clue whether you're understanding or not understanding. Okay. All right. So prophase cell, you're going to have twice the amount of DNA as a normal diploid cell, because the DNA had to be copied to give to the new cell that will be made. So when the new cell is made, it will be diploid. So you go from 12.4 to the second cell, which is 6.2 billion nucleotide pairs of DNA. Okay, so prophase, chromatin condenses into the chromosomes. Okay. And let's see, a couple other things here I'll mention, right? The centrioles, they are going to start moving towards opposite sides of the cell, towards the poles, right? So if you do have access to your digital textbook, look at figure 2.7, look at picture B. I know that those centrioles are really tiny. My old eyes can barely see them comfortably. But if you look carefully in that drawing, you're going to see two tiny pink colored beans. Those are the centrioles. Two pairs of centrioles. They're going to start moving apart towards the opposite ends of the cell. So the two centriole pairs begin to move toward opposite poles of the cell. And of course, we also appreciate that the nuclear envelope breaks down. So here's a question for you. You probably learned in Bio 101 that the nuclear envelope breaks down, but let's think about this with a question. Why is it necessary that the nuclear envelope disappears during prophase, and I could even more generically say mitosis at this point? Why is that necessary, folks? If you think you know the answer, please type that into the chat window. Why does the nuclear envelope have to disappear? Why does that happen? I'm just asking you to dip your toes into this intellectual topic, perhaps a bit differently or more deeply than you have in the past. Okay, so Terrell and Natalie, I really appreciate your responses here. And you know, you're each spot on, right? Think about it. If you look at that picture in your book, the centrioles are outside the nucleus, aren't they? So if the nuclear membrane stays there, there's really no way for the centrioles to connect the spindle fibers to the chromatids. There's no way for the centrioles to connect the spindle fibers to the chromosomes. So there'd be no way for the machinery of the centrioles to move the chromosomes around to support the replication process of the cell. All right, so this is necessary to enable the centrioles, which are positioned outside the nucleus, to attach spindle fibers to the chromosomes. Okay, it's really that simple. If the envelope doesn't disappear, then there's no way you're going to hook up the spindle fibers. It's simply going to be a barrier. Okay, so folks, that is prophase. Please let me know if you have questions. Otherwise, I will just keep right on moving along here. Okay. Next, we have prometaphase. Okay, it's kind of interesting to me, because I've been in this bio game for quite a few decades. You know, when I was learning the cell cycle mitosis, there was no such thing as prometaphase. It was not written in any books. People didn't think about prometaphase. But of course, as society continues to mature and our scientific knowledge, new research comes out, we start to make things more complicated, right? So, okay, prometaphase, you can see in picture C as in Charlie of figure 2.7. The big thing about prometaphase that I'll document with you is that the spindle fibers actually form, they polymerize, and the spindle fibers connect to the chromosomes. Okay, continuing. All right, the next section of mitosis is metaphase. Okay, so with metaphase, there are literally these tiny little, how should I say, tiny little molecular machines that literally walk along the spindle fibers, and they help move the chromosomes into this this relatively straight position in the middle of the nucleus, or where the nucleus was, because the envelope has disappeared. Okay, so typically what we say here is that spindle fibers are used to push / pull, because it can depend on the position of the chromosome, whether you're going to push something kind of away from the central or whether you're going to pull it back closer to the central. Spindle fibers are used to push or pull the chromosomes into a relatively straight line in the interior of the nucleus. Okay, that straight line is traditionally referred to as the metaphase plate, right? I'm sure you've heard that before. Okay, let's finish the note taking here, and then I have, I think, what is a really an excellent video that shows you some of those little molecular machines walking along the spindle fibers. I really want you to see that. But let me just finish the notes here, and that way you'll have them in your brain as we're looking at the video. Okay, so we have metaphase. The chromosomes are moved into relatively the center of the nucleus in a straight line. Then we have anaphase. Okay, during anaphase, these motor proteins that also walk along the spindle fibers, they start to tug apart the chromatids. Okay, they start to tug apart the chromatids. The motor proteins associated along the spindle fibers allow the centrioles to pull the sister chromatids apart. That's what you're seeing in picture E as in egg of figure 2.7. You see that the sister chromatids are in fact being pulled apart. Okay, here's a question for you. Okay, in the chat window, can you answer this question? What is the name of the protein that normally holds sister chromatids together? Okay, that should have come up in your reading. What is the name of the protein that normally holds sister chromatids together? You remember? Okay, all right. Anthony's the first to answer, and he happens to be right, so I'm going to go with that. Good job, Anthony. It is, in fact, cohesins. Okay, so cohesins are the names of the proteins that hold the sister chromatids together. And if you don't remember that, just take a look at figure 2.8. Okay, and then I want to ask you another question. What is the name of the enzyme that breaks cohesin proteins down so that sister chromatids can separate? What is the name of the enzyme that breaks cohesin proteins down so that sister chromatids can separate? This is something that we have not talked about during our lecture time. Anybody know from a previous course? Okay. Thanks, Megan. Thanks, Terrell and Jade. Yeah, Terrell, I like how you think, because it tells me, you know the basic bio, right? ASC's got to be an enzyme. That's fantastic. Okay, so it turns out, Jade, good job, that this enzyme is called separase. Separase is the name of the enzyme that nibbles on the cohesin proteins at the very beginning, or you might say the very end of metaphase, very beginning of anaphase, and breaks their bond between the chromatids so that the spindle fibers can pull them apart. Okay, good job. All right, so separase is the name of the enzyme that does that job. Okay. Let's continue here. All right, so we start separating the sister chromatids, and then, we move into arguably the last stage of mitosis, which is called telophase. All right. Okay, and so you can see a picture there for yourself. With telophase, the separated sister chromatids, they arrive at their new positions along the poles of what will ultimately become two separate cells. The sister chromatids reach separate poles of the cell. Okay. You'll notice from last time, the last lecture we gave, that this cartoon here has cytokinesis as a topic sitting outside mitosis. Your genetics textbook says that cytokinesis should be considered part of telophase. Honestly, I've seen it communicated a different way in different authoritative texts. So I would say probably even the experts don't really agree on this. Okay, so I'll just say this. Telophase, the sister chromatids separate, they go to each respective pole. As that is occurring, cytokinesis will begin. And cytokinesis involves the separation of the cell's cytoplasm. Right? And so if you're an animal cell, you're going to develop what's known as a cleavage furrow that goes basically through the interior middle cytoplasm of that figure 8 cell you see in picture F. If you're a plant cell, well, it's going to be a little bit different because you got a cell wall there. So you're going to make this structure called a cell plate. Okay. But basically, cytokinesis means separation of the cell's cytoplasm. Okay, when cytokinesis completes its job, you should have two physically separate cells that are, in fact, genetically identical. Okay, all right, so let me ask a question. Let me ask a question here in your notes. Okay, when cytokinesis is complete, how many nucleotide pairs of DNA will each of the separated cells have? What do you think, folks? Okay. All right. Very good. Absolutely right, Jade. You're going to be back to your normal 6.2 billion nucleotide pairs of DNA for a diploid cell. Absolutely, yes. Okay. Very nice. Okay. Okay, gang. So any questions at this moment? Okay, so you run through a topic like this in 30 minutes, you say, oh, it seems pretty straightforward. I get this. It seems pretty simple, right? But that kind of does disservice to this topic because, as I've said previously, there's still a tremendous amount about this process we do not understand, and there's still a tremendous amount about this process that, when it in fact goes wrong, leads to cell-based disease states such as tumor formation and then ultimately even cancer. So as much as we do understand, and as eloquently presented in a textbook as it might seem, that, oh, this is, we got this, we get it, we understand it, I think nothing really could be further from the truth. There's still a lot of mystery about things with the cell cycle, with mitosis. So I want to show you that second video here that I think you might enjoy. Okay, so the second video is called Chromosome and Kinetochore. It's by an institute that comes out of Australia, WHAI Movie. So let me put this in your notebook here, and you can take a look at it potentially again on your own study time. But let's take a look at this. It's not going to go through everything of the cell cycle, but it might present some information differently than you've maybe seen it before. Okay, so let's have a look here. And here we go.
DNA in each one of your cells is just 2 nanometers across, but around 30 to 40 million nanometers long. To keep DNA organized and regulate access to the genetic code, it is wrapped around structural proteins to form nucleosomes, which are then further organized into fibers called chromatin. The chromatin fibers are packaged into gigantic molecular structures called chromosomes. We are now pulling away from a chromosome, out through a nuclear pore, which is a gateway through the nuclear membrane. We are zooming back to reveal a cell about to undergo mitosis. The nuclear membrane breaks down, releasing the chromosomes. The chromosomes are arranged in the middle of the cell before being pulled apart, with identical sets of DNA going to each side. The cell then splits down the middle, creating 2 new daughter cells. This visualization isolates a single chromosome at mitosis. The chromosome contains 2 identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle, used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
Okay, folks, that's the end of that particular video. And let me highlight, once again, this channel on YouTube with the WEHI movies, of which this is one video in that series, is really fantastic. They have a lot of great videos. Excuse me. Excuse me. They have a lot of great videos about important biology topics. So, you might, in those geeky moments, you might just want to watch a couple extra ones, okay? But at any rate, let me ask you, okay, in the chat window. Let me ask you in the chat window, okay. Have you ever seen a video of the kinetochore that showed such detail? Have you seen something like that before? I mean, I hope you have, but if you haven't, well, hopefully you enjoyed seeing that. So have you seen something like that before? Okay. All right. So what I'm saying is, as our understanding of these processes deepen, that allows graphic illustration companies that focus on scientific content to make some just amazing, breathtaking videos. And hopefully you saw towards the end section there, you saw those little dynein motors and stuff that were walking along the microtubules of the spindle fibers, right? So there's these little machines walking along those microtubules that are communicating information between the chromosomes and the centrals and saying, okay, we are now ready to separate, or we are not yet ready to separate, okay? Obviously, there's no telephone in there, right? So you literally need to have a molecule go walk back and forth between the centrioles and the proteins that are part of the kinetochore plate attached to the centromeres of the chromosomes, communicate information about when separation should take place during anaphase, okay? It's really, really an amazing process that Mother Nature has somehow created through evolution, okay? All right, thoughts or questions? Okay, well, if you don't have any thoughts or questions about mitosis, then let's talk about the cell cycle and meiosis, okay? So obviously, there is a lot of similarity, but there's also some important distinctions, okay? Okay, so let's see here how I should do this. Okay, let's transition gears here and talk about the cell cycle, okay, interphase plus meiosis. Okay, so we remember that the purpose of meiosis is to intentionally cut the chromosome number in half, and of the chromosomes we do transmit to our children, our progeny, those chromosomes should be genetically diverse and different from the original parental chromosomes. Okay, so, all right, let me see here. I don't want to step through literally everything that I tried to step through with mitosis because I don't think that would be a good use of time. But let me say a couple things here, okay, to set some background, okay. So first off, the stages of interphase that we talked about for the mitotic cell cycle, G1, S, G2, as far as science can tell, those stages are largely intact and largely identical in a cell that's going to perform meiosis. Now, as soon as I say that, of course, somebody's going to come out and say, yeah, but we just found this difference. So I suspect there are some differences, but to a large approximation, we're not aware of any significantly large differences. Okay, so the stages of interphase, G1, S, and G2, that are part of a meiotic cell's cycle are believed to be very similar to the same stages in a mitotic cell cycle. Okay, and by that I mean, cells that are going to potentially perform meiosis still have the ability to leave the cell cycle and go through a G0 period of cell cycle arrest. That can still happen. There are multiple cell cycle checkpoints positioned throughout interphase that we shared with you during mitosis. Cell cycle checkpoints still exist. G0 still exists. Okay. So let me just document that. G0 cell cycle arrest can still take place. And cell cycle checkpoints also still exist to monitor the integrity of the processes that are about to unfold. Okay, we want to make sure that we're ready to copy the DNA. We want to make sure that the DNA has been faithfully copied before we start the first cycle of meiosis 1, right? So meiosis, you have meiosis 1. Meiosis 1, you might have a little brief period, which is another interphase, and then you might go through meiosis 2, right? So it depends on the kind of cell that we're talking about. Different cells on the planet do things a little bit different, right? Not everybody does things the same way. Okay, so meiosis typically has 2 stages, okay? Meiosis 1 and meiosis 2. And these stages might in fact be separated by a brief time of non-nuclear processes, which would represent a miniature interphase 2, okay? These stages in some cells are separated by an interphase 2 period. Okay. So, I'm saying these things, you don't have to get worked up about them. The big goals, that I want you to know for this course is that you really understand meiosis 1 and meiosis 2. I'm not going to care too much if you appreciate anything about interphase 2. I'm telling you it exists, but it's not a really big deal for us here in this course. Okay. So, so as you have been looking at it, right, figure 2.7 in your digital textbook shows you mitosis, and then both figure 2.9 shows you a comparison of mitosis and meiosis, which can be very helpful, and which I will highlight some of those differences between mitosis and meiosis in your notes. And then figure 2.11 kind of really goes through the meiotic stages, meiosis 1, meiosis 2. Can be very helpful to look at these things, and some of the things I challenge you with in today's lab will start to hint at some of these things. So, okay, let's talk some more about meiosis. Okay, Meiosis has two stages, and as we've said, at the end of the second stage, you will basically have 4 cells that are now haploid. You know, keeping with the idea that we're talking about a human cell in the human body. So you're going to start meiosis. You will begin meiosis with a normal diploid sex cell. It's going to be a diploid stem cell that's designed to go through this process, grow and reproduce once, grow and reproduce twice, and end up with 4 haploid gametes, sperm or theoretically unfertilized eggs. Okay, so meiosis begins in a sex stem cell that is diploid, right? It's diploid. It's 2n equals 46. Okay, and through the 2 stages of meiosis 1 and meiosis 2, we end up with 4 haploid cells. After 2 cytoplasmic rounds of division, 4 haploid gametes are produced. The chromosomes in those gametes are genetically diverse. Okay, so let's talk a little bit more about this genetic diversity business. Okay. Sources of genetic variation in meiosis. Okay, so we have 2 sources of genetic variation in meiosis. Okay, the first is probably the most famous of which, and I'm sure you've heard of it. I mean, you've had Biology 101, you've heard of it. This is the crossing over business. Okay, crossing over takes place during prophase 1 of meiosis 1. Okay, so in crossing over, non-sister chromatids of homologous chromosomes will swap genetic cargo, exchange positions. Now, the molecular events underlying these genetic swapping of DNA is unbelievably complicated, and we will broach the complexity of that process in a future chapter. But for right now, we're just going to accept the fact that DNA does exchange position between the non-sister chromatids of the homologous chromosomes. Okay, so crossing over takes place during prophase 1, and during crossing over, segments of DNA between non-sister chromatids, segments of DNA between non-sister chromatids, which are part of homologous chromosomes, swap position. Okay, so I have a question in the chat window. Question in the chat window. Okay. During mitosis, are sister chromatids genetically identical? Okay, good job, folks. Megan, Jelena, Jade, Terrell, Anthony, good job. They absolutely are, right? Sister chromatids in mitosis are genetically identical the whole way through the cell cycle, unless an unusual mutation has occurred, which can happen, leading to tumor formation and cancer and all that ugliness. Okay, next question. Are sister chromatids genetically identical in a G2 cell that will enter into meiosis 1? Okay, all right. So hopefully you can come to, the correct conclusion yourself, and the answer is yes. Nicely done. Next question. Okay. Are sister chromatids in metaphase 1 of meiosis 1 genetically identical? So you can see what I'm driving towards, right? They are in fact not genetically identical because of this crossing event, which happened during prophase 1. When prophase 1 has completed its job, the sister chromatids that have synapsed as part of the homologous chromosome pairs, they're going to swap genetic information, so they can't be genetically identical anymore. Okay. All right. Okay. Another question in the chat window. Here we go. Are homologous chromosomes genetically identical? Okay, so we have one answer. How about the rest? Okay. Okay, very good, folks. The answer is no, right? The homologous chromosomes cannot be genetically identical. If you think about their source, one copy of each pair of homologous chromosomes came from mom, the other came from dad. So they're not going to contribute molecules which are genetically exactly the same nucleotide for nucleotide, right? So homologous chromosomes are not genetically identical, but they are genetically similar because they will contain alleles for the same genes, right? So you might have, you might have an allele for brown eye color on mom's chromosome, and you might have a corresponding allele, for blue eye color on dad's chromosome. Okay, good. I think you're with me, and I appreciate that. So crossing over takes place during prophase 1. Segments of DNA between non-sister chromatids, which are part of the homologous chromosomes, will swap position. We'll talk about the molecular dynamics of that at a later stage. The second part of this genetic variation, and I know we're getting close on our time, but let me just say this. The second part is really the random alignment of the homologous chromosomes. So, it's kind of tough to do this without being in front of you and with the whiteboard and looking at it. But when you look at figure 2.11 in your textbook, okay, I'm going to ask you a question in figure 2.11 of your textbook. Hold on a second, folks. I got to make my language as unambiguous as possible. Okay, hopefully this will be clear. So if you're looking at your textbook, in figure 2.11, metaphase 1, what is the color of the first chromosome on the most top left side of the cell? Most top left side of the cell, What is the color of that chromosome? Okay, good. Hopefully everybody can see that it's blue. And you can see that its homologous chromosome underneath it is kind of like an orangish brown color. And what I'm trying to tell you is there is no reason that the blue chromosome has to be on top and the orange one on the bottom in any arbitrary cell. Well, the orange chromosome could have been on top and the blue one could have been on the bottom. So the arrangement of how those homologous chromosomes order themselves with respect to each other along the metaphase plate is entirely random. It's like flipping a coin toss, whether you get the top one being blue or the top one being orange. That is random. And obviously, which chromosome is there will determine which cell it ends up with after the cytoplasm separates itself, right? So the second point of variation is the random alignment of the homologous chromosomes along the metaphase plate. Okay, the random alignment of the homologous chromosomes along the metaphase plate. Okay, so folks, I know we're out of time, and I promise I'm going to stop now. So here's my goal. Here's my goal. I'm going to continue to lecture in lab at 2:30 for about 30 minutes to really get this chapter well underhand so that we can probably start the next chapter next week. Okay, so I'm going to continue lecture for 30 minutes, wrap up meiosis, then we'll go into our lab time activities that we need to do. So so we'll take a break at this time. And let me just mention, Jade, if you could stay on with me, I have a small email problem I need to talk to you about. Otherwise, everybody else, go take a nice break, come back at 2:30, and we'll see you for lab, okay? Okay. Jade, are you there with me?
Yes, I'm here.
Hi, how you doing?
Good, how are you?
I'm okay. Hey, Jade, I just wanted to mention to you that earlier today, I attempted to send an email to you in response to a Mathematica notebook you shared with me. And when I sent it, the email bounced back to me from Microsoft Outlook, and it said the delivery has failed. So let me share my screen and actually show you what this looks like.
Okay.
Okay, so yes, I wanted you to know first off that I responded and told you that you earned full credit on your first lab, Intro to Mathematica. And I sent it to this address, gxr64441@hack.edu.
Yeah, that's
And
the one I use.
for reasons I do not understand, Microsoft complained about it. So I just wanted to alert you that we might have an issue, but I don't understand what the issue is.
Okay.
Okay.
I wonder if it's still, I wonder if maybe I'm still connected to email from my previous college that we also use through Outlook. I'm not sure if there's maybe something sitting up there that's allowing the connection to be interrupted.
Well, let me ask you this. When you signed up for Mathematica, what email address did you create your Mathematica account with?
Let me go into my account information. I did use my hack email for that one.
Yeah, okay. Well, in that case, I don't know what has happened here. So let's just keep track of this. Like if you're sending me stuff over the next week or two and you're not getting any messages from me back after a few days when you would expect it, there might be a problem.
Okay.
And so let's just keep track of this. Hopefully this was just an odd thing and everything will take care of itself moving forward, okay?
Okay. Just a question. Have you sent any other emails to the class since, I mean, I did receive the initial email where you gave us all the Zoom links and the information for signing into everything. Have you sent any additional emails outside of that potentially haven't received?
No, there shouldn't have been, any classified emails. Any subsequent communication would have been of documents and mastering genetics and stuff. So
Okay.
I don't think you missed anything.
Okay. Do you think if I contacted, like, do they have a technology support at HAC?
They do. I can show you that. Let's see here.
Because potentially I can contact them and ask if there's an issue.
Okay. Let's see here. Yes. Let's do hack.edu. If I go to the main page, if you go to Learning Commons at the top center of the page, you'll get a bunch of support services. Here's our telephone number for IT support, 717-780-2570.
Okay.
So,
Thank you.
so hopefully this is not really an issue, but I just wanted to alert you in case, things start to go off the wheels a little bit here.
No, I appreciate you warning me.
All right, I'll see you at 2:30.
See you at 2:30. Thank you.